Biology.
So the next question is asking, we are being told that Petri dish was put in Clinostat, which was kept rotating. So if you know a Clinostat, this is an apparatus which rotates in order to cancel out the effect of auxin hormone. As you can see, this is a sample Clinostat which is rotating, and as you can see, we have during the experiment we have the plants are being rotated, but after the experiment you can see that the plants have increased, yes, in length, but they have only increased in length going horizontally. But for the control experiment whereby the Clinostat was not rotating, you can see that all the shoots are facing upwards like that. So a Clinostat is an apparatus which is used to cancel out the effect of auxin hormone. Because remember, where auxin hormone is found, that part of the plant is going to grow faster. Now for the Clinostat, as it rotates, it evenly distributes the auxin within the plant. That is why now this plant is going to grow only horizontally in the Clinostat. But for the control experiment, again remember, for the control experiment, the Clinostat is not rotating, so auxins are going to come on the lower side for the shoot, and then the shoots are going to grow faster facing upwards. So in this question we are being asked that a Petri dish was put in a Clinostat which was kept rotating. Explain the results after 2 days. So for the Clinostat, remember again we have said that this is an apparatus which rotates in order to cancel out the effect of auxin hormone and gravity on the seedlings. So this Clinostat is mainly used to check geotropism. So for the geotropism, we will study geotropism in Form 4, the topic of reception and coordination, as you had said. So geotropism is the growth curvature of the plant in response to gravity. Because for the auxins in the shoot side, auxins are pulled on the lower side of the plant, which makes it grow faster than the shoot curves going upwards. So for this Clinostat, we see that the radical grew horizontally, since rotating of the Clinostat evenly distributes auxin hormones in the root region, while in the shoot region also, as the Clinostat was rotating, so there was even distribution of the auxin hormone within the stem, and therefore that's why now the plants also grew horizontally. For the control experiment, remember we say that the Clinostat is not rotating. And since the Clinostat is not rotating, auxins are going to be pulled on the lower side due to gravity. That is for the shoot region, and then it's going to make it to grow, to curve upwards as the shoot is growing. So the next question we are being asked that, I think it's a continuation, we are being asked that the seedling in Petri dish 3 had equal spaced marks on its length at the beginning of the experiment. So the seedling in Petri dish 3. So remember we had 3 Petri dishes, as you can see, Petri dish 1, 2, and 3. So I've been asked that the seedlings in Petri dish 3 had equal spaced marks on its length at the beginning of the experiment. Draw a diagram to show the marks after 2 days. So draw a diagram to show the marks after 2 days. So, like for the marks, you can see that the marks will now be evenly spaced because this will say that the plants have grown. So for the Petri dish 3, remember, if you can look at the diagram, so if you can look at the diagram, you can see that those marks are evenly distributed. But for your answer, these marks now should be spaced. Why should these marks be spaced? So you are spacing these marks in order to stress the point that there has been growth. Growth has taken place. So since growth has taken place, of course, these marks are going now to be evenly spaced. But now, anytime in biology you have been asked to draw a diagram. So drawing a diagram in biology, remember these people, first of all, they are marking the method of answering before they mark your answer. If you have been asked to draw a plant cell or an animal cell under a light microscope, so remember, the examiner is not going to mark what you have answered at first. So they are going first of all to check the question. And that's why in biology we have explain, differentiate, describe, state, list. So the first thing which is marked in biology, they mark the method. Even before checking your answers, they mark the method. have you followed the correct method for the question? The question was asking explain. Did you write your answer in paragraph? The question is asking state. Have you stated your points? Now you see in this question you are being told to draw. Since you have been told to draw, the first thing the examiner is going to mark, they are going to mark the rules for drawing. Have you observed the rules of drawing? Now before they check your diagram. So for the rules of drawing, remember, you must use a pencil. Because if you have used a biro pen and they detect that you have used a biro pen, even they don't check your labeling. They just cancel everything and everything is wrong. So the first thing they check is, have you used a pencil? So that is the first rule. The second rule, you must use a continuous line to draw your diagram. The third rule, remember in Form 1 we said you never shade your diagram. So you should never shade your diagram. The fourth rule, you should label your diagram on the right hand side. And then after that, you should also give your label an appropriate heading. So if you are not labeled the diagram, so you should give your diagram an appropriate heading. So if your diagram doesn't have a heading, you're going to lose marks. And then finally, your diagram should be inside a frame. So put your diagram inside a frame and then label the diagram appropriately. Also, a diagram should be large enough to be read, or it should be legible. Don't draw a very tiny diagram. Draw something big, because the bigger your diagram, the less are your chances of making a mistake when drawing that diagram. But if you draw a tiny diagram, the chances of making a mistake are very high. So that is how the diagram should look like. So the spaces, you should at least increase some space between the points in order to show that growth has taken place horizontally. So the next question is asking, explain the importance of cell differentiation. So we have already gone through this, but remember we are doing revision. So explain the importance of cell differentiation, which is cell modification, etc. So you see that modified and specialized cells are able to carry out specific functions in an organism. Therefore, they increase the chances of survival in the organism. So remember we say that cell differentiation is the same as cell modification, is the same as cell specialization, whereby these modified cells are able to carry out now specific functions in the environment, whereby these specific functions in the environment will increase the chance of survival for that organism. Because a sperm cell will increase the chance of survival for the parent organism, because at least now the generation of this parent is going to be increased. The palisade cells, for instance, they're going to store food for the plant, and then if they store food for this plant, now this organism is going to obtain food at will from this stored food from the palisade cells, etc. So specialized cells, they perform a specific function, therefore increasing the chances of survival for now that whole organism. So question next, we are being told differentiate between primary and secondary growth. So differentiate between primary growth and secondary growth. So what is primary growth? So simply, primary growth, this leads to the elongation of the plant height. So if the plant increases height vertically, that is primary growth. But if the plant increases its width, if the plant increases its width, this is now secondary growth. So in primary growth, this is the increase in height of the plant, while in secondary growth, this leads to the increase of the girth or the width of the plant. So remember again, increase in the height of the plant, this is primary growth, and then increase of the girth in the plant, this is secondary growth. So what are the importance of primary growth? So the importance of primary growth, remember, primary growth mainly takes place in the meristematic tissues of the plant, whereby, remember, we said that we had the apical meristems, we had the lateral meristems, we had the intercalary meristems, as you can see this diagram. So in the meristematic regions, this is now where growth mainly occurs in the plant. In the meristematic regions, again, remember we say that this is where we have auxin hormones, whereby the function of auxin hormone, remember we say that it leads to growth of that part of the plant. So what is the importance of plant growth? So the importance of plant growth, we can say that it enables the shoot to grow upwards in order to absorb sunlight for photosynthesis. So the shoots are able to grow upwards in order to absorb sunlight for photosynthesis. Apart from that, we can also talk about the roots. So It also enables the roots to grow downwards in the soil for support, anchorage, and also absorb nutrients. So for the shoot, they grow upwards in order to absorb maximum sunlight. For the roots, they grow downwards in order to absorb nutrients and also for optimum anchorage of the plant inside the soil. So this diagram again is the diagram of the meristems. It at least shows the different meristematic regions in the plant, whereby, remember, we say that the function of these meristems is to lead to growth. Whereby in this meristem, remember, we said we have auxins. Auxins are hormones which lead to growth. So the next question we are being asked, say the importance of secondary growth in plants. So state the importance of secondary growth in plants. So for the secondary growth, it mainly takes place in these regions which are called the cambium regions. So for these cambium regions, so the cambium regions are mainly found on the stem. So on this stem, we have the cambium regions, whereby the function of cambium is to increase the girth or the width of the stem. That is it. So for the secondary growth, you can see that it enables the plant to increase in girth and width of the stem, thereby providing additional support for the plant. So that is the importance of secondary growth, to increase the girth and the width of the stem in order for the plant to be able to receive additional support. So for the next question, it's asking, use the diagram below to answer the questions that follow. And as you can look at this diagram looks like a lenticel. So the diagram looks like it's a lenticel. This is the diagram of a lenticel. So whereby, remember, we say that for the plant can use the stomata for gases exchange. It can use the lenticel for gases exchange also, whereby stomata is found in the leaves, lenticel are found on the stem, and also we have the pneumatophores. So the pneumatophores, they are found on the root regions for the breathing roots. I guess they are called the hydathodes, the breathing roots, yet to confirm. So the question is asking, use the diagram below to answer the question that follow. This is the diagram, as you can see. Then the first question is asking, name part K to O. So we have K, L, M, N, and O. So the part labeled K, that hole which is found on the stem, which allows respiratory gases to enter or leave, that is, and maybe excess water to enter or leave the stem. So part labeled K, that is called the lenticel. So that is the name given to the part labeled K. What about the part labeled L? So the part labeled L, that is the epidermis, which is now the outermost region of the lenticel. That is the epidermis. And then apart from that, now immediately below the epidermis, this is now the general knowledge for the lenticel. Immediately below the epidermis, we always have the cork cells. So we always have the cork cells immediately below the epidermis, and then immediately below these cork cells, we have the cork cambium cells. So don't forget that we have the lenticel, yes, the lenticel. But now we have the epidermis. Below the epidermis, we have the cork cells. Below the cork cells, we have the cork cambium cells. And then now, below the cork cambium cells, which is now the region O, we have the cortex region. As you can look at this diagram of the lenticel, they reinforce these points that we have just talked about. Because as you can see, that diagram of the stem, those small holes, the small brown holes that you can see, they look horizontal in this diagram because different plant species have different lenticel. But remember, a lenticel, these are the small holes or the small openings which are found on the stem. The small holes or the small openings found on the stem are the ones which are called lenticel. In this diagram, we have those two small brown ones. So the function of the lenticel, remember, they are to allow they are to allow respiratory gases to pass in and out of the stem, as well as excess water to leave the stem region. So that diagram, that was the diagram of the lenticel. So the follow-up question is asking, state the function of the region K, N, and O. State the function of the region K, N, and O. So for K, remember, this is the lenticel. So for the lenticel, we have said that this is the site of passageway for the different respiratory gases to either enter or leave the stem. And then apart from that, we have region N. So for the region N, we see that it gives rise to the cork cells and the secondary cortex, which will now lead to the secondary growth. Secondary growth, remember, we say that it increases the width or the gap of the stem. That is secondary growth. So what about region labeled O? So for the region labeled O, we see that it contains the parenchyma cells, which are used for storage. Also, apart from parenchyma cells, it also contains the collenchyma cells and the sclerenchyma cells, whereby these cells, we study them in the topic of cell specialization subtopic, and then we say that in cell specialization, these are normal plant cells which have been modified to perform the function of support. That is the parenchyma, collenchyma, sclerenchyma. Normal plant cells modified to perform the function of support. So apart from that, these parenchyma cells, now they serve two functions. They can either store photosynthetic product, or also they can be used for support as well. So let's now go to the next question apart from that. So question next is asking, state the plant growth hormones used in plant growth and development. So state plant growth hormones used in plant growth and development. So which are these growth hormones? Already we have labeled, we have auxins. We have said we have gibberellins also. There are many more. So we have ethylene, we have florigen, we have chromatin, we have abscisic acid, we have cytokinin. I think we are going to look at this in the upcoming subtopics, but these are a few of the growth hormones that we have. Ethylene, florigen, gibberellin, cytokinin. We have talked about auxins, which lead to primary growth, etc. So the follow-up question is asking, state any two functions of growth hormones listed in A above. So state two functions of the growth hormones. So the functions of each of these hormones. So like for instance, we have ethylene. So for the ethylene, we see that it leads to the shedding of leaves. Like for example, we can talk about the best plant is the jacaranda tree. During summer, the jacaranda tree sheds off its leaves, and then when we are approaching the rainy season, now the leaves again begin to grow back. So ethylene hormone, it leads to the shedding of the leaf. Not only in jacaranda, but in different plants, in all the plants, we have ethylene. Apart from that, we have the other function, you can say that it promotes fruit ripening. Like for instance, you have ever seen these bananas. So today the banana is green, and then after that, tomorrow the banana has changed yellow, and then like on the next day, the bananas have started to rot. If you press the bananas, they are soft. So what some farmers do is that they inject different fruits with ethylene hormone. So if you inject any fruit with ethylene hormone, that fruit is going to ripen and mature very fast. That is what they do to mangoes, they do to bananas, they do to oranges, etc. So ethylene, remember, it promotes fruit ripening. Apart from that, it also prevents stem height elongation. So if you inject ethylene on a plant, it is going to make the stem to grow very slowly, because the function of ethylene is to prevent stem height elongation. So apart from that, you can say, if now this stem is going, the height of this stem is going to be limited, it is going to make now this stem to grow faster, or it is going to make this stem to thicken faster. So remember, it is going to prevent this stem from growing height-wise, but it is going to encourage this stem to grow width-wise. So the stem is going to thicken much, and then the height is going to be prevented, etc. Among other functions of ethylene. We have many more. So apart from that, you can talk about florigen. So what's the function of florigen? So florigen, it comes from flowers. So florigen, if a flower has florigen, the flower is going to mature very faster. So it promotes flowering in plants. That's the first function of florigen, to promote flowering in plants. Apart from that, it regulates flower development, and also it controls flowering through seasons. That's why in rainy seasons, maybe you can see that flowers are not being produced. But when the rainy season ends, you realize that flowers are being produced. It is the florigen hormone which controls flowering in different seasons of the plant. Among others, let's go to the next hormone, which is not the chromatin. So for the chromatin hormone, you see that it enables the plant to heal and repair. Picture the platelets in the body. So in our body we have platelets. If you have been injured, the platelets are going to heal and repair the wound. That is in human beings, in mammals. But now in plants, we have now this chromatin hormone. So this chromatin hormone is now the hormone which enables healing and repair of the injured part of the plant. Apart from that, you can also see that it enables the plant to resist pathogens. So if there are different pathogens in the environment, this chromatin is also going to enable the plant to resist the different pathogens which are found in the environment. So the other hormone is auxin hormone. So for this auxin hormone, we see that the function of auxin hormone is that, first of all, we say that it facilitates primary growth for the plant. We have said that time and time again. So that is the main function of auxin hormone. But what are the other functions of auxin hormone? So you can see that auxin induces parthenocapy process. But what is parthenocapy? So for this parthenocapy, remember we say that this is the development of a seedless fruit. So whereby now the fruit is going to develop away from or off from the normal process of reproduction. So the development of a seedless fruit, that is what is known as the parthenocapy. Apart from that, you can say that auxin also causes apical dominance. So for this apical dominance is what we said that now the apical meristems, remember, the apical meristems are found on the top side, on the shoot, and on the root region. So for this region, which is called the apical meristem, for the shoot side, it facilitated the plant to grow upwards, for the root downwards. So for this point, apical dominance is that the plant is going to grow faster going upwards and faster going also downwards, and not increasing in girth. That is what we are referring to as now the apical dominance. So the plant is going to grow going upwards and downwards for the shoot and the root, and not grow going sideways, increasing in girth. That is what we have called the apical dominance. So for the auxin, we see that it also promotes ovary development of the flower. It also promotes ovary development. Apart from that, you can say that it also promotes plant to increase in height. So apart from auxins, there are many more functions of auxins, but for now ni hizo. So apart from auxins, let's look at the gibberellin hormones. So for the gibberellin hormone, you see that they end bud dormancy. Apart from that, you can say that they also prevent growth of adventitious roots of the plant. As you can see this diagram, this is the diagram of the adventitious roots, whereby for these adventitious roots, we study them in science, I think science in primary school. So whereby you say the function of the adventitious roots is that these are roots that form from non-different roots patterns. So they are used for absorbing they are used for absorbing water and mineral salts from different regions of the soil apart from the normal roots, and also some of them can be used for the anchorage of the plant. But the main function of the adventitious root is to absorb water and mineral salts apart from the normal roots of the plant. So those are the adventitious roots, which are roots developing from a non-root tissue. If roots develop from the stem, those are adventitious roots. So remember, the adventitious roots, they are roots which develop from non-root tissue. Those are the ones which are now called the adventitious roots. So apart from that, let's briefly talk about the abscisic acid. So the abscisic acid, we see that it inhibits the shoot growth. So that's the first function of abscisic acid. It inhibits the shoot growth. Apart from that, it inhibits the development of side branches in plants. Apart from that, it also inhibits stem elongation and promotes leaf fall. It promotes leaf fall, whereby in biology this we called it abscission. So it promotes leaf fall, which in biology we called it abscission process. So what about cytokinin or cytokinin? So for the cytokinin, we see that it promotes cell division in the meristematic region. whereby in the meristematic region, remember we said we have the apical meristems found on the shoot and apical meristems found on the root region. Also, we see that it inhibits cell elongation, and then it also promotes stem thickening. Yes, it also promotes stem thickening apart from promoting shoot formation, among others. There are also different functions of the cytokinin, but those are just a few, but we have even more functions of the cytokinin. So we go to the next question. It's asking, define the term apical dominance. So what is apical dominance? Remember this diagram yenye tumepitia time and time again. So what is apical dominance? So apical dominance, we have defined it previously, but also again we are revising, so we can say this is the inhibition and prevention of the lateral branches from growing, which prevents the formation of side branches. So in apical dominance, the height-wise of the plant has dominated growth. The plant is going very fast, going upwards and downwards, and then it has prevented growing sideways of the stem. That is apical dominance. The meristematic regions have dominated growth. They are growing faster than the side buds. That is what is called apical dominance. Whereby in apical dominance, the meristems dominate growth, hence the name apical dominance. As you can look at this diagram, the meristematic regions. The meristematic regions, the apical meristematic regions, they are growing faster than the side, they're growing faster than the other meristems found in the stem, like the cambium. So they are accelerating the process of growth, they are growing faster. So for the next question is asking, where is the question? The next question is asking, name the hormone associated with apical dominance. Of course, the hormone is auxin hormone. That is the hormone which is mainly associated with apical dominance to facilitate the plant to grow ikienda juu na ikienda chini. Whereby we see that auxins accelerate the growth of shoot and the root tips. So it facilitates also rapid cell division in these regions, apart from cytokinin venye tumeona. It facilitates rapid increase and growth in cells and cell division, thereby making these regions to even grow faster than the other meristems. So the next question is asking, say the importance of apical dominance. Apical dominance, we have mentioned it a million times. So the importance of apical dominance is to increase the height of the plant. That is it. That is the only function of the apical meristems, is to increase the height. Remember, height ni juu chini, height ni vertically. This is the width. Function of the apical meristematic region is to increase the height. The roots to go downwards and the shoot to go upwards. That is it.
Biology.